In this video, I'm going to talk about the neuromuscular junction. And essentially, this is a junction or a connection between a neuron, which is essentially a working cell of the nervous system, and a skeletal muscle cell. That's the connection between these two. Okay, so let's start with a neuron first. Talk about the basic anatomy of a neuron. So I'm going to draw a really basic neuron up here. Now neurons have a few major components. They have this major big portion up here that's going to have all the organelles and go through all the metabolic processing. This part of the cell is called the cell body or the cell soma. All right. And you have processes that come off of the cell body or the cell soma. You have typically much smaller processes, these little processes that make it kind of look like a star. These are the dendrites. And dendrites are pieces of the neuron that allow electrical signals to come into the cell body. So keep in mind that neurons work off of electrical principles. They use electrical signals very similar to electricity running through electrical wires in the walls. So when signals come into the cell, they travel through dendrites. Okay. Now neurons also have a very long process coming off of the cell body called an axon. And axons are used to carry signals away from the cell body. Okay, so carry signals away from the cell body down towards the end of the axon, this little bulb-like region right here called the axon terminal. So that's the end of the axon. Okay. Now the axon terminal of a neuron can form what's called a synapse with many different types of cells. A lot of times they synapse with other neurons, so it may look something like this, where you have another neuron kind of sticking off the end there. But in this case, when we're talking about the neuromuscular junction, what you're actually going to see synapsing here, forming a connection, is a skeletal muscle cell. So I'm just going to draw a little skeletal muscle cell. Right? Remember, it's multinucleate and it's striated. So here's a little skeletal muscle cell. Now the junction between the axon terminal and the skeletal muscle cell, this whole junction here, is called the neuromuscular junction, or NMJ. Okay, that's the connection between these two. So I'm going to blow up the neuromuscular junction a little bit and draw it down here. So let's start with the axon terminal. So that's what I'm going to be drawing right here. So I have my axon, and here's that bulb-like axon terminal at the very bottom. Okay, so I just flipped it around just to make it a little bit easier to draw, so it's pointing down instead of side to side here. And then what I'm drawing on the bottom is going to be the sarcolemma of the skeletal muscle cell. Okay, so this is the muscle cell here. This is the axon terminal here. Okay, now the reason I drew these weird wonky looking folds on the sarcolemma, these folds are called junctional folds. And their purpose is to increase the surface area of this cell membrane. Okay, now we have receptors on this cell membrane that I'll get to in just a second. And so the more folds you have on the membrane, the more receptors you can fit on that membrane. Increases the surface area for those receptors. Okay. Now in the axon terminal, we have little vesicles called synaptic vesicles. So this is a synaptic vesicle. And synaptic vesicles hold chemicals called neurotransmitters. Okay, so that's these little dots I'm going to put in here. These are neurotransmitters. Now you can have different neurotransmitters in different neurons. It's going to depend on what the neuron is, what kind of cell it's targeting, as to what type of neurotransmitter is there. Okay, but in this case, since we're targeting skeletal muscle cells specifically, the neurotransmitter is going to be a special kind called acetylcholine, or I'm just going to abbreviate it as ACH. Okay, so we have acetylcholine inside of these synaptic vesicles, and their whole purpose is to get released into this synaptic cleft here, where they will end up binding to these receptors, okay, and causing a reaction. So we'll get to that in just a second. So the very first thing that's going to happen if you're trying to trigger one of your muscle cells to contract is your neuron, okay, will send a signal down along the axon. So it'll send a signal along this axon, an electrical signal, okay? And as soon as that electrical signal makes its way to the axon terminal, which is what's happening here, there are channels on the axon terminal. These are calcium channels on either side that when electricity hits them, they're what we call voltage gated channels. So when the electricity hits them, they're going to open up. Right? So they're going to open up in this membrane. You're going to put a nice hole in the membrane specific to calcium. Okay, and we have a lot of calcium floating around outside of our neurons. So what is going to happen through diffusion once these channels open up? Well, the calcium, let me erase a little bit of this just to clear up any confusion here. Once we open up these channels, all right, this calcium that's floating around outside will start to diffuse into that axon, excuse me, into the cell. And when calcium diffuses into the axon terminal, that causes these little synaptic vesicles, these little bubble-looking structures, to start to move down. They'll move down towards the bottom of the axon terminal, and they're going to start fusing with the axon terminal. Okay? They're going to start fusing with the axon terminal. And when they do that, they're going to be releasing their contents, which again are those neurotransmitters, more specifically that acetylcholine. Okay, and acetylcholine will diffuse across that cleft, and it'll start binding to these receptors on the skeletal muscle cell. All right, and when that happens, the next thing that's going to happen is those channels will open up. Right? They'll open up in the membrane. And these channels are specific to sodium especially. They're really ion channels, okay, which remember an ion is any charged atom. But the major ion that's going to be involved here is sodium, okay? So let me see if I can move down a little bit, try to clear up some space. So sodium, you are going to have a lot of, so let me just draw this membrane again just to get a little bit more space, okay? So if your membrane looks like this, all right, it's the same membrane we got right above. I'm just trying to make a little more space. And we have a lot of sodium floating around outside of our muscle cells and not very much inside of the cell. What's going to happen if you open up these channels on this membrane? Well, the sodium is going to start to diffuse into the cell, okay? And what happens is the inside of the cell starts to become a little more positive. The outside charge starts to change to a little bit more negative. And this whole process, we'll get more into this in the nervous system chapter, but this process is called depolarization. And the depolarization event is really what's going to trigger that muscle cell to contract. Okay? There's a little bit that goes on after the depolarization, but that is the event that tells the muscle cell, okay, it's time to contract. This is time to go. Now, after we are done with the contraction, what needs to happen to all of this acetylcholine that's hanging out in this synaptic cleft up here? Remember, that's what's making the channels open, right? That's what's making the depolarization happen. So what do we need to do with that acetylcholine? We got to get rid of it, right? If we don't want our muscle to contract anymore, we got to get this acetylcholine out of here so we're not stimulating it anymore. And so there are a couple ways we can do this. One, we can either take the acetylcholine back up into the axon terminal and then just recycle it and use it again later. Or two, we have enzymes called acetylcholine esterase that are like little Pac-Mans kind of, that come in and eat up all the acetylcholine that's found within that cleft, and they digest it and remove it out of that gap, which, by the way, this little space between these two structures is called the synaptic cleft. All right, synaptic cleft. So keep that in mind. So again, acetylcholine esterase is an enzyme that cleans up the synaptic cleft by removing all of those acetylcholine neurotransmitters, okay? Or the neurotransmitters can just kind of diffuse out of the cleft. And if they're not down here in this cleft, in this space, then you really don't have to worry about them. They're not going to be affecting the muscle cell at that point, all right? So this picture down here is a better picture of a neuromuscular junction, so you can see things a little bit more clearly. Again, you see the synaptic vesicles here. Let's see if I can try to highlight them. So you got the synaptic vesicles right there. You've got the neurotransmitters inside of those synaptic vesicles, more specifically acetylcholine. Down here we have the acetylcholine receptors colored in purple, and you have those junctional folds on the sarcolemma of the skeletal muscle cell, and again, those are to increase the surface area. All right, and you can't see the calcium channels in this image, but those would be, right around the surface of this of this axon. Okay, and so those are going to be allowing calcium to get into that axon terminal, and that's what will trigger these neurotransmitters to move down and fuse with the end of the axon terminal and release those acetylcholine neurotransmitters. Those neurotransmitters will diffuse across that synaptic cleft. They'll bind to their receptors. That will cause those acetylcholine receptors to open in the cell membrane of the muscle cell. And when those open, keep in mind that sodium will diffuse into that cell, and that will cause a depolarization event. Okay, depolarization. That depolarization event will eventually trigger that muscle cell to contract. Okay. Now, neuromuscular junctions are seen in every single individual skeletal muscle cell in your body. Every skeletal muscle cell in your body has its own neuromuscular junction. So it is an extremely intricate structure and setup. All right. All right, so y'all look over the neuromuscular junction and let me know